We're going to be starting on chapter 18 now. We're going to be looking at the cardiovascular system, specifically the heart in this chapter. So we're going to be looking at the anatomy and physiology of the heart. We're also going to learn about the cardiac cycle and the various factors that influence heart rate and also the force of contraction of the heart. Now the heart is relatively small. It's roughly the same size as your closed fist, and it rests on top of your diaphragm near the midline of the thoracic cavity. Now recall that the midline is that imaginary vertical line that divides the body into equal left and right sides. Now the heart lies in the mediastinum. This is an anatomical region that extends from the sternum to the vertebral column, back here. And it also extends from the first rib to the diaphragm and between the lungs. About 2/3 of the mass of the heart lies to the left of the body's midline. You can see most of it is here near that left lung. You can kind of visualize the heart as a cone lying on its side, the pointed end here and kind of like this. Well, sort of. Now, the pointed apex of the heart is formed by the tip of the left ventricle, the lower chamber of the heart, and it lies on the diaphragm. The base of the heart is opposite the apex and it in its posterior aspect. And the base of the heart is formed by the atria of the heart, the upper chambers, mostly by the left atrium. Now here we can see our outer and inner layers of the serous pericardium that make that double layer serous membrane around the heart. So here we have this pericardial cavity that is the inside of the serous pericardium, and it's kind of like a little fist punching into a limp balloon. You can kind of think of the heart as the fist here, and then we have these two layers of the balloon that kind of surround the heart. So remember that interior layer that touches the organ of a serous membrane is going to be that visceral layer. So here, this layer, this interior layer that surrounds and touches the heart is going to be the visceral layer of the serous pericardium. And then the exterior layer here is going to be the parietal layer of the serous pericardium. So that pericardium confines the heart to its position in the mediastinum, while allowing sufficient freedom of movement for a lot of vigorous and rapid contraction. So remember that pericardium is going to be that membrane that surrounds the heart. Now the pericardium itself consists of two parts. It's going to be the fibrous pericardium and the serous pericardium. Now the deeper serous pericardium is a lot thinner and more delicate membrane that forms a double layer around the heart. And that serous pericardium itself has two layers. So that last slide that we just looked at, that was the serous pericardium. We have that outer parietal layer, which is also fused to the fibrous pericardium. And then the inner visceral layer of the serous pericardium, which is also called the epicardium, is one of the layers of the heart wall, and it adheres tightly to the surface of the heart. Now, the wall of the heart consists of 3 layers. There's the epicardium, which is the external layer, the myocardium, the middle layer, and the endocardium, which is the innermost layer. So the epicardium is composed of 2 layers. The outermost layer of the epicardium is called the visceral layer of the serous pericardium. So there's kind of some overlap in the pericardium and the layers of the heart. So the outermost layer of the epicardium is also the visceral layer of the serous pericardium. Now, the epicardium contains blood vessels, lymphatics, and vessels that supply the myocardium. So we see those here. The middle layer of the heart wall is the myocardium. This is responsible for the pumping action of the heart, and it's made up of cardiac muscle. This is also the thickest layer of the heart wall, makes up about 95% of the heart wall. And then the innermost endocardium. This is a thin layer of endothelium that overlays a thin layer of connective tissue. Really, it just provides a smooth lining for the chambers of the heart, and it also covers the valves of the heart. In this picture here, we can also see that fibrous pericardium that we kind of talked about in the last couple slides as one of those pericardial layers. Now, the heart has 4 chambers. The superior 2 chambers are the atria. So we have a right atrium and a left atrium, which is kind of in the back here, but it's kind of back there. And then the 2 inferior pumping chambers are the ventricles, right ventricle and a left ventricle. So the paired atria receive blood from the blood vessels returning blood to the heart, called veins, while the ventricles eject the blood from the heart into blood vessels called arteries. So the atria receive blood from veins, and the ventricles send blood to arteries. So these are going to be 2 terms that we need to get used to. Veins bring blood to the heart, and arteries take blood away from the heart. Those are going to be the definitions of those 2 words. Now, on the anterior surface of each atrium is this wrinkled pouch-like structure that's called an auricle. So we can see the auricle here for the right atrium, and the auricle for the left atrium is there. Now, these little wrinkled structures here, kind of here and here, these are named auricles because they resemble a dog's ear. Auricle means ear. And we'll talk about their purpose a little bit later. So on this picture here, this is the posterior view of the heart. We can see the left atrium there, the right atrium there. And also see that left and right ventricle. Now, those auricles that are above the atriums, each of those auricles slightly increases the capacity of an atrium so that it can hold a greater volume of blood. Also, on the surface of the heart are going to be a series of grooves called sulci, or singular is sulcus. And these grooves of the heart contain the coronary blood vessels, and they're usually covered by fat. So here we have this posterior interventricular sulcus that's covered by fat. And there's going to be the coronary sinus that is found within that coronary sulcus there as well. So those little grooves that have a lot of blood vessels in them. Those coronary blood vessels, supplying blood to the heart muscle directly. So here we can see a nice anterior view of a frontal section now. So we're looking at some internal anatomy in the heart. The right atrium forms the right surface of the heart, and it receives blood from three veins. The superior vena cava, which is here, the inferior vena cava, which is down here, and also the coronary sinus. Which is going to be coming through the posterior end, but you can kind of see the opening here. Remember that veins always carry blood towards the heart. Now the anterior and posterior walls of the right atrium are actually quite different from one another. Inside of the posterior wall, it's quite smooth, but inside the anterior wall is really rough, and this is due to the presence of these muscular ridges called pectinate muscles. So again, those pectinate muscles are going to be found on the anterior wall of the atrium, or the right atrium. And these pectinate muscles also extend into the auricle. Between the right atrium and the left atrium is this thin partition that's called the interatrial septum. It's going to be behind here, behind this large pulmonary trunk, but that's the interatrial septum. That separates the left from the right atrium. Septum, this word means a dividing wall or a partition. Now blood passes from the right atrium into the right ventricle through a valve that's called the tricuspid valve. And it's called a tricuspid valve because it consists of three cusps, or also known as three leaflets. Now this valve is also has another name too. It also is called the right atrioventricular valve. And this is because it's a valve between the right atria and right ventricle. Now these valves of the heart are composed of dense connective tissue that's covered by endocardium. The right ventricle is about 4 to 5 millimeters in average thickness of the ventricular wall, and it forms most of the anterior surface of the heart. Now the inside of the right ventricle contains a series of ridges formed by raised bundles of cardiac muscle fibers, which are called trabeculae carnae. Now these are labeled in the left ventricle, but we see the trabeculae carnae in the right ventricle as well. Some of the trabeculae carnae actually convey part of the conduction system of the heart. Now the cusps of the tricuspid valve, remember that's the valve between the right atrium and the right ventricle, the cusps of this valve are connected to tendon-like cords, which are called chordae tendineae. So the cusps are attached to the chordae tendineae, and those chordae tendineae in turn are connected to cone-shaped trabeculae carnae, which are called papillary muscles. So these papillary muscles themselves are going to be those cone-shaped trabeculae carnae. So the cusps are attached to the chordae tendineae, which are attached to papillary muscles. Now internally, the right ventricle is separated from the left ventricle by a partition called the interventricular septum. Separates those two ventricles. Now it's not really labeled on here, but about right here is going to be another valve. So I'll maybe label it up here. This is going to be the pulmonary valve. This is also called the pulmonary semilunar valve. And blood will pass from the right ventricle through this pulmonary valve into a larger artery called the pulmonary trunk. And the pulmonary trunk divides into right and left pulmonary arteries and carries the blood to the lungs. So again, it's not really labeled on this particular picture, but this pulmonary trunk, about right here, is going to divide into right and left pulmonary arteries. So the right pulmonary artery is going to go behind here, and the left pulmonary artery goes out here. And these are going to carry blood to the lungs. Remember, arteries always take blood away from the heart. So pulmonary arteries will take blood from the heart to the lungs. Moving on to the left atrium. The left atrium has walls that are about the same thickness as the right atrium, and the left atrium forms most of the base of the heart. It receives blood from the lungs through 4 pulmonary veins. So here we have the left pulmonary veins and the right pulmonary veins. So remember, veins bring blood to the heart. So this is blood coming from the lungs. Actually, in this picture here, we can see those pulmonary arteries that branch off from the pulmonary trunk. So just kind of briefly going back to the right ventricle, we can see this pulmonary valve here. See this pulmonary valve that leads blood from the right ventricle into the pulmonary trunk. And then here we can actually see those branches off of the pulmonary trunk here, that left pulmonary artery, And over here, this blue colored one is going to be that right pulmonary artery. So this is a really nice picture diagram that we can see those arteries taking blood from the right ventricle and taking it to the lungs. Okay, coming back to talking about the left atrium now. So right inside the right atrium, it's a really quite smooth posterior wall, because we can kind of see here, look at the wall of the left atrium here. It's very smooth. I'll erase these circles here. But in the left atrium, very, very smooth, especially when compared to the right atrium here. We could see definitely a lot less smooth. Within this right atrium here, we can see all those pectinate muscles. So remember, the pectinate muscles give the right atrium the look of this kind of roughened surface. These are muscular ridges made by the pectinate muscles. So you can definitely see those differences between the left and the right atrium, that left atrium having very smooth surface. Now the pectinate muscles are actually confined to the auricle of the left atrium, and this is why the anterior wall of the left atrium is going to be smooth. Now blood passes from the left atrium into the left ventricle through the bicuspid valve. This is also called the mitral valve. And as the name implies, it has two cusps. Bicuspid is two cusps, just like the tricuspid valve between the right atria and right ventricle has three cusps because it's tricuspid. So that blood is going to pass from the left atrium into the left ventricle through that bicuspid valve. And this is also going to have that other name, also going to be known here as that left atrioventricular valve. So it kind of has two names. One of the name refers to the actual structure of the valve, the bicuspid valve, because it contains two cusps. And the other name has to do with its function. So it is the passageway of blood from the left atria to the left ventricle, hence the left atrioventricular valve. Here we can see an inferior view of a transverse section of the two ventricles. So on the left of our view here is the right ventricle, and then on our right of this picture is the left ventricle. So the left ventricle is the thickest chamber of the heart. It averages about 1,015 millimeter thickness, and it forms the apex of the heart. Now, just like the right ventricle, the left ventricle contains trabeculae carneae and has chordae tendineae that anchor the cusps of the bicuspid valve to the papillary muscles. Now, blood passes from the left ventricle through the aortic valve and goes from the left ventricle into the aortic valve, or through the aortic valve rather, and then into the ascending aorta. Now, some of the blood that enters the aorta flows into coronary arteries. Coronary arteries. And these will branch from the ascending aorta and carry blood to the heart wall. So the coronary arteries will actually supply the heart itself with blood that it needs for nutrients and to get rid of waste. Because the heart itself is its own organ, it has muscle cells, it has endothelial cells. So all these cells also need their own blood supply to bring oxygen and nutrients and to take away waste and carbon dioxide. And this is all completed by the coronary arteries. Now the blood in the ascending aorta that doesn't go to the coronary arteries will go through the arch of the aorta and then into the descending aorta. Branches of the arch of the aorta and the descending aorta will actually carry blood throughout the whole body. Now, in addition to cardiac muscle tissue, the heart wall also contains dense connective tissue that forms the fibrous skeleton of the heart. Now, essentially, the fibrous skeleton consists of 4 dense connective tissue rings that surround the valves of the heart, and they actually fuse with one another and merge with the interventricular septum. So we can see these rings here, these rings around the valves. So here we have the pulmonary valve, and then we have the pulmonary fibrous ring. This is part of the fibrous skeleton of the heart. We have the aortic valve and the aortic fibrous ring around it. There's also the bicuspid valve, and then the ring around the bicuspid valve. Remember, that's also known as the left atrioventricular valve. So this is the left atrioventricular fibrous ring. And then same over here. Tricuspid valve and the right atrioventricular fibrous ring. Now, in addition to forming the structural foundation of the heart valves, the fibrous skeleton prevents overstretching of the valves as the blood passes through them. It also serves as a point of insertion for bundles of cardiac muscle fibers, and it acts as an electrical insulator between the atria and the ventricles. So very important function of that fibrous skeleton as well. As each chamber of the heart contracts, it pushes a volume of blood into a ventricle or out of the heart into an artery. Now, valves will open and close in response to pressure changes as the heart contracts and relaxes. Each of the 4 valves helps to ensure one-way flow of blood by opening to let blood through and then closing to prevent backflow. So this view down here in the bottom, this is a superior view of our atrioventricular and semilunar valves. So here we can see the bicuspid valve, very large valve, and also the tricuspid valve are also quite large. Because they're located between an atrium and a ventricle, the tricuspid valve and the bicuspid valves are termed the atrioventricular valves. because they go from the atria to the ventricles. So up here, we can also see the bicuspid valves. When an atrioventricular valve is open, so like on this left picture here, the rounded ends of the cusps will project into the ventricle. So the ventricle is going to be in this portion here. The bottom and the atria is on the top here. So you can see the cusps will project down into the ventricle here. Now when the ventricles are relaxed, the papillary muscles are also relaxed. Then the chordae tendineae are slack. So the chordae tendineae are up here. They are slack, and the blood will move from a higher pressure in the atria into a lower pressure in the ventricles through those atrioventricular valves. So here we're going to have high pressure because the atria is full of blood, and the ventricle is going to be low pressure because there's going to be little to no blood within the ventricle. So they're moving from high pressure to low pressure, so the blood will naturally move down into the ventricle when the valve is open. Now, on the right here, when the ventricle contracts, the pressure of the blood will drive the cusps upward until their edges meet and they close the opening. So this is where the ventricles contract. Now at the same time that the ventricles contract and the pressure of the blood drives those cusps upward to close, at the same time the papillary muscles contract, which pulls on and tightens the chordae tendineae. So it's kind of being pulled from here, but even though it is being pushed by that backflow of blood. So these tight chordae tendineae prevents the valve cusps from everting. So it keeps them from kind of opening up back into the atria. And it is possible that they would open up due to this really high ventricular pressure, but the really tight chordae tendineae keep that from happening. They keep the cusps closed and tight against one another, and it keeps them from everting, opening into the atria. Now, if the atrioventricular valves or the chordae tendineae become damaged, blood flow may regurgitate or flow back into the atria when the ventricles contract. So that's when the cusps themselves or the valves or the chordae tendineae are damaged. They don't hold the cusps closed proper when the ventricles contract. So that's in a damaged heart. Now the aortic valve and the pulmonary valve, these are also known as the semilunar valves. And this is because they're made up of these 3 crescent moon-shaped cusps, each of them are. So you can kind of see like here, 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 and same with that pulmonary valve. These 3 crescent moon shapes. So semilunar. Now each of the cusps attaches to the arterial wall by its convex outer margin. The semilunar valves allow for ejection of blood from the heart into arteries, but they prevent backflow of blood into the ventricles, just like we saw before with preventing backflow. The free borders of the cusps will project into the lumen of the artery. Now, when the ventricles contract, the pressure builds up within the chambers, within those ventricles. This is when the semilunar valves open, and the semilunar valves will open when pressure in the ventricles exceeds the pressure in the arteries. And this allows the ejection of blood from the ventricles into the pulmonary trunk and the aorta. One other really important thing of note is that when one set of valves is open, the other set is closed. So on this left picture here, the bicuspid valve and the tricuspid valve, those atrioventricular valves, when they're open, you can see the pulmonary valve and aortic valve, the semilunar valves, are closed. On the right here, when the semilunar valves are open, the atrioventricular valves are closed. So very important thing there. After birth, the heart pumps blood into two closed circuits with each beat. These are the systemic circulation and then the pulmonary circulation. Now these two circuits are going to be arranged in series. So what that means is that the output of one becomes the input of the other, and the output of that one is going to become the input of the first one. So this would be the same thing that would happen like if you connected two garden hoses front to back to each other. Now the left side of the heart is the pump for the systemic circulation. And then the right side of the heart is the pump for the pulmonary circulation. So on the left picture here, we can see a full diagram of the heart and where blood pumps. And this right picture kind of shows those two circuits and how they connect to one another. So on the right side of this picture here, we have the left side of the heart. So like I said, this left side of the heart is the pump for the systemic circulation. So it's going to receive bright red oxygenated blood or oxygen-rich blood from the lungs up here. So the lungs are going to send this bright red oxygen-rich blood through the pulmonary veins to the left atrium, and then the blood will flow through the bicuspid valve to the left ventricle, and then the left ventricle is going to eject blood into the aorta through the aortic valve. From the aorta, blood divides into separate streams, entering progressively smaller systemic arteries. And these systemic arteries will carry the blood to all the organs throughout the body, except for the air sacs of the lungs or the alveoli of the lungs. These are going to be supplied by the pulmonary circulation. So those systemic arteries will get smaller into systemic capillaries at the organs. And this is where we have the transfer of oxygen and carbon dioxide within those capillaries around the organs. Now the right side of the heart is that pump for the pulmonary circulation. So the right side of the heart receives all of the dark red deoxygenated blood, receives it returning from the systemic circulation. So the vena cavas as well as the coronary sinus are going to pump that deoxygenated blood. Now that it's lost its oxygen to the organs, it's going to pump it back to the right atrium. The blood is going to pump from the right atrium to the right ventricle through the tricuspid valve. And then the blood ejected from the right ventricle pumps or flows into the pulmonary trunk, which branches into pulmonary arteries that will carry blood to the left and right lungs. Now, in the pulmonary capillaries, the blood will unload its carbon dioxide and pick up oxygen from the inhaled air. And then this freshly oxygenated blood will then flow into the pulmonary veins, return to the left atrium, and then we're beginning the cycle once again. So that's kind of a very simplified, broken down cycle of the systemic and pulmonary circulation. Systemic circulation just means the circulation of the whole body, systemic meaning body. So this is going to be the circulation that goes to all the organs of the whole body, muscles, everything else. That's going to be down here. Pulmonary circulation just means to the lungs. Now on this picture on the left, we essentially see the same thing, but we see it directly through the picture of the heart. And the numbers you can see here are going to line up with the pictures, or the numbers on the right picture. So here we have number 1 on the heart is the right atrium here. And so essentially we could just follow the same path through the numbers that we just followed with the right picture. So right atrium to the right ventricle to the pulmonary trunk. Number 4 is going to be those pulmonary capillaries to the lungs, both sides. Then number 5 here on the right and left side of the heart are going to be those pulmonary veins bringing oxygenated blood to the heart back from the lungs. Enters into the left atrium, then to the left ventricle, and then to the aorta and systemic arteries at number 8. Number 9 is the systemic capillaries. The ascending aorta is going to bring blood to the systemic capillaries of the head and upper limbs. And the descending aorta down here is going to bring blood to the systemic capillaries of the trunk and the lower limbs. And then here we have the inferior vena cava and the superior vena cava bringing blood back to the right atrium. Now the nutrients are not able to diffuse quickly enough from the blood in the chambers of the heart to supply all the layers of cells that make up the heart wall. So because of this, the myocardium has its own network of blood vessels, and this is called the coronary circulation, also known as the cardiac circulation. Now the coronary arteries, these are going to branch from the ascending aorta, and they're going to encircle the heart, kind of like a crown encircles the head. Now, while the heart is contracting, little blood flows into the coronary arteries because they're kind of squeezed shut. When the heart relaxes, however, the high pressure of the blood in the aorta will propel blood through the coronary arteries into the coronary capillaries and then into the coronary veins. So when the heart is contracting, coronary circulation, we could say, is closed. And then when the heart is relaxing, coronary circulation is open. Now there are two coronary arteries, the left and right coronary artery, that will branch from the ascending aorta. And they're going to supply this oxygenated blood to the myocardium. The left coronary artery passes inferior to the left auricle, and it divides into the anterior interventricular, so this interior interventricular branch, and it also is going to divide into the circumflex branches. So we have the left coronary artery will branch into the anterior interventricular branch. Let's say anterior interventricular branch, and then also the circumflex branches. So the circumflex branch is right here. Now that anterior interventricular branch, this is the artery in the anterior interventricular sulcus, and it supplies oxygenated blood to the walls of both ventricles. The circumflex branch, this lies in the coronary sulcus, and it distributes oxygenated bloods to the walls of the left ventricle and left atrium. Next, the right coronary artery. This supplies small branches to the right atrium. So you can see this here. Now, it continues inferior to the right auricle, and ultimately it divides into the posterior interventricular and marginal branches. So we have the posterior interventricular branch and the marginal branch here. Now the posterior interventricular branch is going to follow the posterior interventricular sulcus of the heart, and it will supply the walls of the two ventricles with oxygenated blood. So it's kind of helping to supply the posterior part of the ventricles, whereas the anterior interventricular branch is going to supply the anterior parts of the ventricles. Now the marginal branch beyond the coronary sulcus is going to run along the right margin of the heart, and it's going to transport oxygenated blood to the wall of the right ventricle and right atrium. Now, after blood passes through the arteries of the coronary circulation, it's going to flow into capillaries where it delivers oxygen and nutrients to the heart muscle, and it also collects carbon dioxide and waste, and then that blood moves into the coronary veins. Remember that transfer of oxygen, carbon dioxide, nutrients, and waste can only occur at the capillaries. Transfer of these molecules can't occur in arteries or veins at all. It only is going to occur in capillaries. Now, most of the deoxygenated blood from the myocardium will drain into a large vascular sinus in the coronary sulcus on the posterior surface of the heart, which is called the coronary sinus. So this is where most of that deoxygenated blood drains into. The deoxygenated blood in the coronary sinus will empty into the right atrium. Now, the principal tributaries that carry blood into the coronary sinus are going to be the great cardiac vein. This is in the anterior interventricular sulcus, which drains the areas of the heart supplied by the left coronary artery. So it drains areas supplied by the left coronary artery. The middle cardiac vein, actually we can probably write this over here. So I'll do different colors, sorry about that. So these are going to be the main veins bringing blood to the coronary sinus. The great cardiac vein is our first one. It's going to drain the areas of the heart supplied by the left coronary artery. Oops. Next one is going to be that middle cardiac vein. These are all going to be veins, by the way. The middle cardiac vein is in the posterior interventricular sulcus, and it will drain the areas supplied by the posterior interventricular branch of the right coronary artery. So I'll just put the areas here that it's going to drain from, or it'll drain the areas that are supplied by these arteries that I'm going to write out. So posterior interventricular branch. Our third major vein of the coronary circulation is the small cardiac vein. This one is right here. And this is in the coronary sulcus, and it's going to drain the right atrium and the right ventricle. So this is going to be pretty much areas of the blood, or areas of the heart, supplied by the marginal branch. That marginal branch of the right coronary artery. Because remember, the marginal branch is going to give oxygenated blood to the right ventricle and the right atrium. lastly is the anterior cardiac veins. So that is found here. And this is going to drain the right ventricle. And this is actually going to be the vein that opens directly into the right atrium. So all of the other veins kind of all go through the coronary sinus into that anterior cardiac vein to drain directly into the right atrium. So this one doesn't directly correlate with an artery, so I am just going to put draining the right ventricle. The others all kind of directly correspond with a coronary artery, and so they're going to take that deoxygenated blood directly from those areas supplied by specific arteries, but the anterior cardiac vein is kind of the last vein in the series that brings all of that deoxygenated blood back to the right atrium. Now let's look at cardiac muscle tissue itself a little bit. Now compared with skeletal muscle fibers, cardiac muscle fibers are going to be shorter in length, and they're also going to be less circular in transverse section. They also exhibit branching, and this gives individual cardiac muscle fibers this stair-step appearance. Usually there's going to be one centrally located nucleus in a typical cardiac muscle fiber, although occasionally a cell may have two nuclei. Now the ends of cardiac muscle fibers will connect to neighboring fibers by these irregular transverse thickenings of the sarcolemma called intercalated discs. Now within these discs, they have desmosomes, which will hold the fibers together, and we'll also find gap junctions within the intercalated discs. And the gap junctions allow muscle fiber action potentials to conduct from one muscle fiber to its neighbors really quickly, almost instantaneously. So gap junctions allow the entire myocardium of the atria or ventricles to contract as a single coordinated unit. The mitochondria are a lot larger and also more numerous in cardiac muscle fibers than in skeletal muscle fibers. In cardiac muscle fiber, they take up to like 25% of the cytosolic space. In a skeletal muscle fiber, only take up about 2% of the cytosolic space. Cardiac muscle fibers have the same arrangement of actin and myosin within their sarcomere. So we have those thin filaments, remember that actin, and the thick filaments made of myosin. They have the same bands, A bands and I bands, the same zones, and the same Z discs as the skeletal muscle fibers do. The transverse tubules of cardiac muscle fibers are wider, but they're also less abundant than we see in skeletal muscle fibers. An inherent and rhythmical electrical activity is the reason for the heart's lifelong beat. The source of this electrical activity is a network of specialized cardiac muscle fibers that are called autorhythmic fibers, also known as pacemaker cells, and they're called these autorhythmic fibers because they are self-excitable. Autorhythmic fibers repeatedly generate action potentials that trigger heart contractions. This picture here shows how the cardiac action potentials will propagate through the conduction system. So we kind of have this numbered sequence like we've seen in other pictures. So starting here at number 1, this is where cardiac excitation normally begins at this place called the sinoatrial node. Now this is located in the right atrial wall, just inferior and lateral to the opening of the superior vena cava. So this vein here. Now sinoatrial node cells do not have a stable resting potential. Rather, they repeatedly depolarize to threshold spontaneously. Now, the spontaneous depolarization is known as the pacemaker potential. Now, when the pacemaker potential reaches threshold, kind of like we saw in that other picture on the last slide, it's going to trigger an action potential. Now, each action potential from the SA node, or the sinoatrial node, will propagate throughout both atria. So you can see these arrows here. We're going to propagate here, but also propagate through the other atria. And it's going to travel by gap junctions. Remember, those gap junctions are found in the intercalated discs of the muscle fibers. So the action potential from the SA node will propagate throughout both atria via gap junctions in the intercalated discs of the atrial muscle fibers. So following the action potential, the two atria will contract at the same time. So at number 1, both atria contract at the same time. Now, by conducting along atrial muscle fibers, the action potential reaches this next location at number 2, which is called the atrioventricular node, also known as the AV node. And this is located in the interatrial septum. And this is just anterior to the opening of the coronary sinus. Now, at the atrioventricular node, the action potential slows considerably as a result of various differences in cell structure in this node. So this is where we have the action potential slows down. Now this delay provides time for the atria to empty their blood fully into the ventricles. Now from the atrioventricular node, the action potential will enter the atrioventricular bundle. So this is at number 3. The AV bundle, which is also known as the bundle of His. This bundle is the only site where action potentials can conduct from the atria to the ventricles. So it's going to be this little area here where that action potential travels from the atria to the ventricles. So action potential travels from atria to the ventricles. After propagating through the bundle of His, the action potential enters both the right and left bundle branches. The bundle branches extend through the interventricular septum. So this septum between the two ventricles is going to travel down that interventricular septum toward the apex of the heart down here. So the action potential travels down the interventricular septum to the apex of the heart. Now finally, at the apex of the heart, we have these really large diameter Purkinje fibers. And these are going to be axons that rapidly conduct the action potential beginning at the apex of the heart towards the remainder of the ventricular myocardium. So these Purkinje fibers will propagate the action potential up through the rest of the ventricles. So Purkinje fibers are very large diameter. Remember, this is one of the characteristics that increases action potential propagation speed. So because of that, they're able to really increase the speed of the action potential so that we can get that action potential really quickly through the rest of the ventricle. So these Purkinje fibers are going to very rapidly conduct that action potential through the remaining ventricles, or through the remaining parts of the ventricles. And this is starting at the apex. So once the action potential propagates through these Purkinje fibers, the entire ventricles contract, pushing the blood upward towards those semilunar valves. So when the ventricles contract, it pushes blood up to the semilunar valves. So this is essentially how an action potential propagates through the entire heart to initially get the atria to contract, and then down here to get the ventricles to contract. Those autorhythmic fibers of the sinoatrial node, or the SA node, these are going to be the natural pacemakers of the heart. Now they fire an action potential about 100 times a minute on their own. So they'll fire action potentials 100 times a minute. So this is what those autorhythmic fibers would naturally like to do on their own. But there's going to be nerve impulses from the autonomic nervous system and blood-borne hormones like epinephrine that can modify the timing and strength of each heartbeat, but they don't establish the fundamental rhythm. The action potential initiated by the SA node will travel along that conduction system that we just talked about, and it spreads out to excite the working atrial and ventricular muscle fibers, and these are called the contractile fibers. So the contractile fibers are going to be fibers of the atrial and ventricular muscle that are actually going to do the pumping, actually going to do the contracting of the heart. So in a contractile fiber, this is how an action potential occurs, this picture on the left. So at number 1, we have our depolarization phase. So this is very different from those autorhythmic fibers that we just looked at. The contractile fibers kind of have this different type of action potential, or how it works essentially. So unlike the autorhythmic fibers, contractile fibers do have a stable resting membrane potential, and it's going to be close to -90 millivolts. Now, when a contractile fiber is brought to threshold by an action potential from neighboring fibers, its voltage-gated fast sodium channels will open. Now, these channels open even quicker than our normal sodium channels that we've seen, say, in nerves. Now, the inflow of sodium down the electrochemical gradient produces this really, really rapid depolarization. Within a few milliseconds, the fast sodium channels will automatically inactivate and the sodium inflow decreases. This is when we kind of get to our next step, which is our next phase, I guess, is the plateau phase. Now, this is going to be the next phase of an action potential in a contractile fiber. This is the plateau phase. This is a period of maintained depolarization. Now, this part of the action potential is due in part to the opening of these voltage-gated slow calcium channels that open in the sarcolemma. Now, when these channels open, calcium ions will rush into the cell from the interstitial fluid. The increased calcium concentration in the cytosol will ultimately trigger contraction. Now, several different types of the voltage-gated potassium channels are also found in the sarcolemma of contractile fibers. And this depolarization is sustained during the plateau phase because calcium inflow balances out the potassium outflow. So I'll write this up here. Calcium inflow balances the potassium, which is K plus. So calcium inflow balances out that potassium outflow. So that's how we get this plateau or this maintained depolarization phase. And lastly, we get our kind of third step of an action potential in a contractile fiber. Now, the recovery of the resting membrane potential during this repolarization phase here. Now, this really resembles the repolarization phase that we've seen in other excitable cells. So it's going to look very similar in a cardiac contractile fiber. Now, after a delay, which is particularly prolonged in cardiac muscle, additional voltage-gated potassium channels open. Now, so here we're going to get additional outflow of potassium ions, and this is going to restore the negative resting potential. During this period, we're also going to get closure of those calcium channels. So really, we're just going to see this very, very quick depolarization and a prolonged or maintained depolarization in contractile cardiac muscle fibers. In contrast to skeletal muscle, cardiac muscle produces little of the ATP it needs by anaerobic cellular respiration. So instead, it mostly relies exclusively on aerobic cellular respiration in its numerous mitochondria. But like skeletal muscle, cardiac muscle also does produce some ATP from creatine phosphate and some from anaerobic glycolysis. But like I said, majority is going to be completed by aerobic cellular respiration. And this is because it has a ton of those really huge mitochondria, so it uses those quite exclusively. As action potentials propagate through the heart, they generate electrical currents that can be detected at the surface of the body. An electrocardiogram, which is also known as an EKG or an ECG, this is a recording of those electrical signals. On a typical record, there's going to be 3 clearly recognizable waves that appear with each heartbeat. So we see these in this picture here, which is an EKG. Now the first wave that we see is called the P wave. This is a small upward deflection in the ECG. And the P wave represents atrial depolarization. Remember that atrial depolarization is going to spread from the SA node through contractile fibers in both atria. The second wave that we see here is called the QRS complex. So we see this here with the QRS complex here. Now this begins as a downward deflection, so we can see this here, but then continues as a very large upright triangular wave right here into this R segment, and then it ends as a downward wave like so. The QRS complex represents rapid ventricular depolarization. So this entire QRS wave is that rapid ventricular depolarization, and this happens as the action potential spreads through those ventricular contractile fibers. The third wave, this is a dome-shaped upward deflection called the T wave. And the T wave indicates ventricular repolarization. So this occurs just as the ventricles are starting to relax. Now, analysis of an ECG will also involve measuring the time spans between the waves. So these are called intervals. So the first interval that we have is the PQ interval. And this is the time from the beginning of the P wave to the beginning of the QRS complex. And this represents the conduction time from the beginning of atrial excitation to the beginning of ventricular excitation. So from the start of atrial excitation to the start of ventricular excitation. So that's going to be that PQ interval. Then we have the ST segment. So this is going to begin at the end of the S wave, or the end of the QRS complex, and it ends at the beginning of the T wave. So what this represents is the time when the ventricular contractile fibers are depolarized during the plateau phase of the action potential. So during that plateau phase. So that's going to be that ST segment here. And then the last interval that we have would be the QT interval. So this interval extends from the start of the QRS complex, and it goes all the way to the end of the T wave. Now this is the time from the beginning of ventricular depolarization to the end of ventricular repolarization. So to the end. So ventricular repolarization. So that's going to be that QT interval there. The atria and ventricles depolarize and then contract at different times because the conduction system routes cardiac action potentials along a specific pathway. The term systole, this means contraction, and this directly refers to the phase of contraction, where the phase of relaxation is termed diastole. Now the ECG waves will predict the timing of atrial and ventricular systole and diastole. Now at a heart rate of about 75 beats per minute, here we have a specific timing, this picture on the left. So at number 1 at the top here, this is where the cardiac action potential arises at the sinoatrial node, and it will propagate throughout the atrial muscle and go down to the AV node in approximately 0.03 seconds. So very, very quickly. Now, as the atrial contractile fibers depolarize, this is where we start to get that P wave forming in the ECG. This is where we get that action potential in the SA node. And this is where depolarization of those atrial contractile fibers starts, and it produces the P wave. Now, as the P wave begins, this is when the atria contract. We get atrial systole. So it's always going to be depolarization followed by contraction. These don't occur at the exact same time. The conduction of the action potential slows at the AV node because the fibers there have much smaller diameters and a lot fewer gap junctions. So that's really going to be the main reason that the action potential slows at that atrioventricular node. We can even write that here. Slows the action potential. Smaller nerve fiber diameters, and also fewer gap junctions. Now we kind of get this resulting 0.1 second delay from kind of down here actually. So this 0.1 second delay gives the atria time to contract, therefore adding to the volume of blood in the ventricles before ventricular systole begins. Now, the action potential propagates rapidly again after entering the AV bundle. That's where we get to number 3 here. So depolarization is going to progress down the interventricular septum and then upward from the apex and outward from the endocardial surface, and this produces that QRS complex. So our action potential is going to go down the interventricular septum to the apex of the heart and then around both sides of the ventricles. Now at this same time that the action potential is traveling down and around the ventricles, this is when atrial repolarization is also occurring. But it's not usually evident in an ECG because this really, really large QRS complex can kind of mask that atrial repolarization. So here, this is where we're also getting atrial repolarization. If you're kind of wondering, well, we see atrial depolarization here in number 1, but where do we see atrial repolarization in an ECG? And this is where. We We don't really see it on the ECG, and that's because the ventricular depolarization really masks that atrial repolarization. And that's because when the ventricles depolarize, it produces this really huge QRS complex that really covers up anything else that's happening at the moment. So we get depolarization of the ventricular contractile fibers, producing that QRS complex. And then directly after that, we get the ventricular systole. That's the contraction of the ventricular contractile fibers. Now this begins shortly after the QRS complex appears in the ECG. And this ventricular systole is going to continue during the ST segment. Now, as contraction proceeds from the apex towards the base of the heart, so the contraction is going to kind of go this way, up both sides of the heart, from the apex towards the base, the blood is going to be squeezed upward toward the semilunar valves. So blood is pushed, here we'll put this color here, blood is pushed up this way and up this way in the left and right ventricles. At number 5 here, we have the repolarization of those ventricular contractile fibers. This is also going to begin at the apex of the heart and spread throughout the ventricular myocardium. And this is going to produce the T wave. So here we do see the repolarization of the ventricles in our ECG. This is because nothing else is masking it. We can definitely see it, and we see it as a T wave. Now this produces the T wave in the ECG approximately 0.4 seconds after the onset of the P wave. Now shortly after the T wave begins, the ventricles start to relax. So then we get that ventricular diastole, that relaxation. So again, just like we get depolarization that's followed by contraction, in the same way we're going to get repolarization followed by relaxation. So it's going to be the action potential flows through the muscle fibers and that produces contraction. It doesn't happen at the exact same time. Now usually it's about 0.6 seconds that this entire thing takes place. So by 0.6 seconds, the ventricular repolarization is complete and the ventricular contractile fibers are all relaxed. And then we start on to the next cycle. Now, a single cardiac cycle includes all of the events associated with one heartbeat. So a cardiac cycle consists of systole and diastole of the atria, plus the systole and diastole of the ventricles. Now, in each cardiac cycle, the atria and ventricles will alternately contract and relax, forcing blood from areas of higher pressure to areas of lower pressure. So as the chamber of the heart contracts, the blood pressure within that chamber increases. Now we're going to be looking at all of the events that occur within one cardiac cycle. So the electrical events, the pressure changes in the chambers, even the heart sounds, volume changes of the chambers, and the mechanical events that occur. So in this first slide here, we're looking at the electrical events that occur during a single phase of a cardiac cycle. So during our atrial systole, the atria are contracting, and at the same time, the ventricles are relaxed. So at number 1 here, we have depolarization of the SA node causes atrial depolarization. So remember, we get atrial depolarization that begins the P wave in the ECG. So we get depolarization of the atria starting right here, but the contraction of the atria doesn't start until here, where we get the beginning of atrial systole. At number 4 here, the QRS complex in the ECG is going to mark the onset of ventricular depolarization. So again, the electrical event is right here. We get the ventricular depolarization. So I'll just label this VD, ventricular depolarization. And then here in this purple color, we get ventricular systole. So that's going to kind of be right there where we don't get the actual contraction of the ventricles until kind of the middle of the QRS complex. Then at number 8 here, we get the T wave in the ECG, which marks the onset of ventricular repolarization. So kind of our main electrical events that occur when we're looking at an ECG are going to be number 1, atrial depolarization. Number 4 here, which is ventricular depolarization. And then number 8 here, which is ventricular repolarization. So these are going to be our 3 major electrical events that we can see on an ECG. Again, like I said before, the atrial repolarization is really masked on the ECG by this huge QRS complex. Here in this picture, we can see the pressure changes in the heart. So these colors of the lines, we can see the red one is the pressure in the aorta. Blue is going to be the pressure in the left ventricle. So remember, the left ventricle is going to be the thickest ventricle of the heart. It's going to be the part of the heart that pumps blood to the systemic circulation, so pumping blood to pretty much most of the body. So it's really big, has large pressure changes. And then our green color is going to be the pressure in the left atria. So this is just representative of the atrial pressure. So we'll start here at number 2. So remember that atrial depolarization is going to cause that atrial systole here in the pink. So this is at the beginning of atrial contraction. And as the atria contract, they exert pressure on the blood within the atria. And this is going to force blood through the open AV valves into the ventricle. So we're going to get that increase in pressure both in the atria and in the ventricle. See these little bumps here in the pressures. So that's when the atria contract. At number 5, This is where that ventricular depolarization causes ventricular systole. Now, as ventricular systole begins, pressure rises inside the ventricles and it pushes blood up against the atrioventricular valves, forcing them to shut. Now, at this time, all 4 valves are closed, so the ventricular volume remains the same. So this is where we get that bicuspid valve closing, because ventricular systole begins and it pushes the blood up against that bicuspid valve, closing it. So we kind of get this increase in pressure right here within the ventricle. As all 4 valves are closed, we're kind of getting this increase or buildup of pressure in the ventricle as it's starting to contract, but that blood doesn't really have anywhere to go. Number 6 is when the aortic valve opens. So continuous contraction of the ventricles causes pressure inside the chambers to really rise quite sharply. As the aortic valve opens, we kind of get that sharp increase of pressure in the aorta, but the ventricles are still contracting as well, so they also are continuing increase in pressure as well. At number 9 here, this is when ventricular repolarization causes ventricular diastole. And as the ventricles relax, pressure within the chambers falls. And the blood in the aorta and pulmonary trunk begins to kind of flow backward toward the regions of lower pressure in the ventricles. So we kind of get this downward pressure drop in the ventricles. Number 10 here is when the ventricles continue to relax and the pressure falls very quickly. And when the pressure falls below atrial pressure, as we get here, this is when the atrioventricular valves open and ventricular filling begins. So this is where the bicuspid valves open and the atria will kind of passively fill the ventricles with the blood that's in the atria. So the atria are not contracting yet at this point, but the bicuspid valve opens, so just by gravity, the blood starts to fill into the ventricles from the atria. Now this picture shows the times in the cardiac cycle when you hear heart sounds. The sound of the heartbeat comes primarily from blood turbulence caused by the closing of heart valves. This is because smooth flowing blood is usually silent, so it's going to be that turbulent blood that causes noise. Usually our first heart sound is going to be louder and a bit longer than the second sound. S1, this sound is caused by the blood turbulence associated with the closure of the AV valves soon after ventricular systole begins. So this is caused by the closure of the AV valves, those atrioventricular valves. So that's the noise that causes that S1 heart sound. Now the second sound, labeled S2, this is going to be shorter and also not as loud as the first, and it's caused by blood turbulence associated with the closure of the semilunar valves at the beginning of ventricular diastole. So caused by that closure of the semilunar valves. That's what causes that S2 noise. Now this diagram indicates the ventricular volume changes during the cardiac cycle. So here at number 3, we see that the atrial systole contributes about 25 milliliters of blood to the volume that's already in each ventricle. So here we have approximately 105 milliliters of blood that's already in each ventricle here. And so this volume here is about 25 mils, and this is the amount that we get to here that is contributed by atrial systole. Now at the end of atrial systole, this is also going to be the end of ventricular diastole. So this is going to be the end of ventricular relaxation. So each ventricle contains about 130 milliliters of blood at the end of its relaxation period, or at the end of ventricular diastole. So because of that, this blood volume is called end diastolic volume. We also call this EDV. Now at number 7 here, the left ventricle ejects about 70 milliliters of blood into the aorta, and the right ventricle is also going to eject that same volume of blood into the pulmonary trunk. So the volume remaining in each ventricle at the end of systole, about 60 mils, is going to be our end systolic volume, or ESV. So what that means is that our stroke volume, this is going to be the volume ejected per beat from each ventricle, equals the end diastolic volume minus the end systolic volume. So at rest, our stroke volume is about 70 mils. So our stroke volume, this is going to be the volume of blood ejected from each ventricle per beat, per heartbeat. So again, our stroke volume, oops. Sometimes we're going to label this SV also, is going to be equal to our end diastolic volume, EDV, subtracted by ESV, which is our end systolic volume. And then at rest, our stroke volume is going to equal about 70 milliliters. So we already looked at the electrical occurrences that are happening within the heart, but now we're going to look at the mechanical phases. So first, atrial contraction, here at the beginning, this is going to cause the blood to be pumped down to the ventricles. You can see with these black arrows here. Then, when both the semilunar and atrioventricular valves are closed, this period is called isovolumetric contraction. So this is when all the valves are closed. Now, during this interval, the cardiac muscle fibers are contracting and exerting forces, but they're not yet shortening. So it's not an actual shortening contraction. During the next phase, the ventricles are contracting and pushing blood to the systemic and pulmonary circulations. So here. Then after the semilunar valves close, there's going to be a brief interval when ventricular blood volume does not change because all 4 valves are again closed. So this is called the isovolumetric relaxation. So also in this phase, all of our valves are closed. So here, this is where semilunar valves are open, and then they close again. Now the second half of this relaxation period, in this light blue here, this is where the atrioventricular valves are open. And ventricular filling begins. So this is where the ventricles fill with blood coming from the atria. And then the next cycle begins with atrial contraction. Again, kind of bringing us back to the beginning. So here we kind of have this full diagram of all the different parts of the cardiac cycle. At the top here we have the ECG, we have the pressure changes, we have the heart sounds, the ventricular volume, and then each phase of the cardiac cycle, so that you can kind of see how each of these items is related to one another. But again, you do have each of these individually on separate slides, but it's kind of nice to see them all together and see how they relate and how they correspond to one another. Cardiac output, abbreviated CO, is the volume of blood ejected from the left ventricle or the right ventricle into the aorta or pulmonary trunk every minute. So this is going to be an important part of it. Cardiac output is based per minute. So cardiac output is going to equal the stroke volume, which is the volume of blood ejected by the ventricle during each contraction. And we're going to multiply stroke volume by the heart rate, the number of beats per minute. And when we do this multiplication, it will give us the cardiac output, which is the volume in milliliters ejected by a ventricle per minute. Now, a healthy heart will pump out the blood that enters its chambers during a previous diastole. So in other words, if more blood returns to the heart during diastole, then more blood is ejected during the next systole. Now there are 3 factors that regulate stroke volume and ensure that the left and right ventricles pump equal volumes of blood. Our first factor is called preload. So this is the degree of stretch on the heart before it contracts. So degree of stretch on the heart before contraction. Our second factor is contractility. So this is the forcefulness of contraction of individual ventricular muscle fibers. Okay, individual muscle fibers. And then our final factor that kind of regulates or changes stroke volume would be afterload. And afterload is the pressure that must be exceeded before ejection of blood from the ventricles can occur. So pressure that must be exceeded, and this is before. So it's the pressure that essentially the heart has to overcome in order to pump blood from the ventricles. Now, adjustments in the heart rate are really important in the short-term control of cardiac output and blood pressure. The sinoatrial node is going to initiate contraction, like I said, but if it's left to itself, it would set a constant heart rate of about 100 beats per minute. We kind of went over that here. So the sinoatrial node, it would like to set a heartbeat at about 100 beats per minute. This is also just abbreviated BPM. However, tissues require different volumes of blood flow under different conditions. And so because of that, our body has several different ways to regulate our heart rate. So these factors are here, and we'll go over them kind of briefly. Things like the autonomic nervous system, hormones and ions, even things like our age and gender and physical fitness are ways that our body can help to regulate our heart rate. Temperature is also going to be another factor that influences our heart rate. So nervous system regulation of the heart is going to originate in the cardiovascular center in the medulla oblongata. So this region of the brainstem is going to receive input from a variety of sensory receptors from the higher brain centers and from other sensory receptors. So these are going to be things like the cerebral cortex, the limbic system, hypothalamus, but also receiving input from sensory receptors like proprioceptors and chemoreceptors, even baroreceptors. Now that cardiovascular center receiving all this input is then going to direct the appropriate output by either increasing or decreasing the frequency of nerve impulses in both the sympathetic and parasympathetic branches of the autonomic nervous system. So our cardiac accelerator nerves of the sympathetic system are going to be able to increase the rate of spontaneous depolarization in the sinoatrial and atrioventricular nodes in order to increase the heart rate. And it also can increase contractility in order to increase the stroke volume. The vagus nerves, on the other hand, from the parasympathetic branch of the autonomic nervous system, will decrease that rate of spontaneous depolarization of the sinoatrial and atrioventricular nodes in order to decrease heart rate. So this is going to be a major way that the autonomic nervous system can regulate the heart rate to both increase or decrease the heart rate based on a lot of input coming from higher brain sensors and sensory receptors. So a major regulation here. So like I said, things like age, gender, physical fitness, and even body temperature are also going to influence our resting heart rate. So increasing our body temperature as it occurs from fever or even strenuous exercise causes the sinoatrial node to discharge impulses more quickly, thereby increasing our heart rate. Now, decreased body temperature will decrease our heart rate as well as the strength of contraction. Now, this figure here summarizes the factors that can increase our stroke volume as well as increase heart rate, both working to achieve to increase our cardiac output. So things like increasing our preload, ways to increase contractility and decrease afterload, all work to increase stroke volume. And other factors like our nervous system, chemicals, and physical fitness, gender, age, are all going to be under other factors that will work to increase our heart rate and thereby increasing cardiac output. Now, a person's cardiovascular fitness can really be improved at any age with regular exercise. Now, some types of exercise are going to be more effective than others for improving the health of the cardiovascular system. But things like aerobics, any activity that works large body muscles for at least 20 minutes, will elevate cardiac output and accelerate metabolic rate. It can also increase our HDLs, which are high density lipoproteins, which are also known as good cholesterol. Exercise and aerobic exercise will also decrease triglycerides, improve lung function, decrease blood pressure overall, and also assist in weight control. All things to overall increase our cardiovascular fitness. And that will be the end of Chapter 18.